Welcome to the short review of type 4 hypersensitivity, also known as delayed type hypersensitivity. I'm Christopher, and in this video we'll shift away from antibody-mediated reactions and focus on T cells, macrophages, delayed inflammation, and why these reactions often take days rather than minutes. The short version is type 4 is delayed and cell-mediated. Unlike types 1, 2, and 3, type 4 hypersensitivity is not primarily antibody-mediated. Instead, it is driven by cell-mediated immunity, specially sensitized T lymphocytes. When T cells recognize antigen, they release cytokines that recruit and activate macrophages and other inflammatory cells. In some type 4 reactions, cytotoxic T cells may also directly damage target cells. This process takes time. Instead of seconds or minutes, type 4 reactions usually develop over 24 to 72 hours, and some granulomatous reactions may persist even longer. That delayed timing is one of the major clues. One vaccine-relevant pattern is a delayed local reaction at the injection site. Clinically, this may appear as a firm swelling or nodule. Sometimes this reflects granulomatous inflammation. A granuloma is a localized area of chronic inflammation made up largely of macrophages and some other immune cells. It can form when the immune system is trying to wall off or contain something that it cannot easily clear. Type 4 hypersensitivity can contribute to granulomatous inflammation because T cells recruit and activate macrophages. But here's the nuance. Not every granuloma is automatically a type 4 hypersensitivity reaction, and not every post-vaccine lump is a granuloma. The key question is whether delayed cell-mediated immunity is driving the lesion. Many things can cause swelling after an injection. A post-vaccine lump could represent transient inflammation, a seroma, an abscess, irritation, or granulomatous inflammation. In cats, persistent or enlarging post-injection masses deserve special attention because of the concern for feline injection site sarcoma, or FISS. FISS is clinically important, but I do not want you to classify it as a type 4 hypersensitivity reaction. It is better thought of as a serious injection site complication associated with chronic inflammation, and it deserves careful clinical follow-up. For this video, our main immunology point is simpler. Delayed local inflammation often makes us think about T cells, macrophages, and type 4 mechanisms. Type 4 hypersensitivity appears in many settings beyond vaccines. A classic example is allergic contact dermatitis, where an antigen contacts the skin and leads to delayed inflammation after sensitization. This is not atopy. Another classic example is the tuberculin type reaction, where prior sensitization leads to delayed local response after antigen is placed in the skin. Graft rejection can also involve type 4 mechanisms because T cells recognize foreign or altered cell surface molecules and drive tissue injury. The common thread is that the reaction is T cell mediated, delayed, and associated with cellular inflammation rather than antibody or immune complex deposition. A memorable example of type 4 hypersensitivity is poison ivy contact dermatitis. The key molecule is urushiol, which can be found in plants in the sumac family, including poison ivy, poison oak, and poison sumac. Related compounds can also be relevant with plants such as mango and the lacquer tree. Urushiol is small and sticky. By itself, it is too small to be a complete antigen. But when it binds to skin proteins, it creates a new antigenic target. This is the idea of a hapten. We saw the hapten concept before with type 2 hypersensitivity and extrinsic antigens. The immune system is not simply responding to an unmodified self-protein, and it is not responding to the urushiol floating alone. It is responding to a new combined target formed when urushiol binds to skin protein. Because T cells need to be sensitized, a first reaction may take longer to appear. After memory T cells are present, re-exposure can produce the familiar rash within a couple of days. Memory changes the timeline. To summarize, type 4 hypersensitivity is cell-mediated rather than antibody-mediated. It is driven by sensitized T cells, cytokines, macrophage activation, and sometimes cytotoxic T cell injury. It takes time to develop, often 24 to 72 hours, because cells need to be recruited and activated. Common examples include contact dermatitis, tuberculin-type reactions, granulomatous inflammation, and some forms of graft rejection. In skin, antigen-presenting cells, or APCs, such as Langerhans cells, can help initiate the response by presenting antigen to T cells. The key pattern is delayed timing, T cell involvement, and cellular inflammation. Thanks for watching. I'm Christopher, and what you understand today will help you protect patients tomorrow. I'll see you in the next video.